Good morning, everyone. Confirm me, am I audible or not?
Recording in progress.
Just confirm me, am I audible or not? Okay. Let's wait for exactly 1 minute and we'll start our lesson today. Okay. In today's class, we are going to we are going to complete the Porifera and we'll solve some of the questions. Today, we are going to see the skeleton system of Porifera and the reproduction and test with respect to the Porifera. Okay, did you revise? What are all the topics which I have been taught yesterday? Okay, whatever it is in test, everything has been going to show. Okay, let answer for my question. What is the technical term of the pores which is present in entire body of the porifera or sponges? What is the technical term of the pores which is present in the sponges? Austria. Very good. What is this phylum Porifera? It is made up of two different layers, inner layer and outer layer. What is the name of the inner layer and what is the name of the outer layer? Pinacoderm is an inner layer or outer layer? And what is the other name of the other layer? See, it is made up of two different layers, that is inner choanodon and the outer pinacoderm. Okay, and the outer pinacoderm, it is made of, it has such cells, with the cells which is present in the outer layer, which is called as the pinacoderm, is called the, which is sites, which is cells, pinacocytes. Okay, then the cells which is present in the choanodon, that is the inner layer, is called the choanocytic cells, and the flagella which is present in the, which layer means inner layer, choanodon. And what about the middle layer? And the middle layer is said to be as the mesophyll or the mesenchyme. Okay, and the cells which is present in the mesenchyme or mesophyll cells is called the amoebocytes. And what about the skeleton structure? The skeletal structure is made up of two different things, and one is spicules and the spongial fibers. Today we are going to see elaborately about the skeletal system. Okay. And in which class the glass sponges have been placed? Answer for this question, in which class? We have said in Porifera there are three different classes, Calcarea, Desmospongia, Hexactinellidae. In which class glass sponges have been placed? Very good. Hexactinellidae. Good. And the place where the place or the pores which have been connected, the incurrent canal and the radial canal, it is called as the opening, which connects the incurrent canal and the radial canal is said to be as, what is the name of the opening which connects the incurrent canal and the radial canal? Prosophile, very good. And the opening which connects the excurrent canal and the central body cavity, that is spongocele, is said to be as the opening which is connecting. Very good. Apophile, clear. And finally, the water has been sent out from the body of the sponges to the surroundings is osculum. Okay, fine. This is the major parts of the canal system. In yes, osculum. See the canal system. In the last DL exam, the canal system has the question has been asked from the porifera is two. Okay, and one question is from the canal system. After finishing the skeletal system and reproduction system, I'm going to conduct a test. And in that test, I'm going to show what is the PYQ came from the porifera. That too from the canal system. Clear? Okay. And which is there are three different types of canal system we have seen yesterday: ascon type, sycon type, and the leucon type. In that, which is the most simplest type of the canal system? Which is the most simplest type of the canal system? Very good, ascon type. And which is the most complex type of canal system? The most complex system is, oh, very good, leucon type. The most simple type is ascon type, complex type is sycon type, and the most simple, most complex type is leucon type. And what is the name of the larval stage undergone by this leucon type of canal system? The poriferans or the sponges, which has the leucon type of canal system, has which type of larva? What is the name of the larval system? That is why the leucon system is also called as another name. It has another name because of the larval stages it undergone. Very good. Ray gun larva. Clear? Super. Okay, that's all about the questions. Let us skip to the skeleton and sponges. See, as I said before, the skeletal system is made by two things. One is calcareous spicules, calcareous spicules because it is made up of a calcium carbonate, as well as the silicaceous spicules is also there, and another one is proteinaceous spongin fibers. Okay, these two things you have to remember. What are all the things? With respect to the skeletal system of porifera, two things. One is it is made up of the calcareous spicules or the silicaceous spicules, and the second one is it is made up of a spongin fibers, that is proteinaceous spongin fibers. Clear? And another thing is what are all the cells gives rise to this spicules, and what are all the cells which gives rise to the spongin fibers? In yesterday's class I have explained you that, and answer me for this question: which cells gives rise to spicules and which cells gives rise to the spongin fibers? Quick, Anjaneyulu, answer for this question. Which cell, for example, the inner layer coinoderm is made up of coinocytic cells. Okay, the outer layer pinacoderm is made up of the pinacocytic cells. Like in the same way, what is this cell gives rise to the spicules and what is the cells gives rise to the spongin fibers? Lalita, answer for this question. These are all the things where the question have been arised frequently. See, the scleroblast, it is in the general characteristics. If you revise those general characteristics, means you could have easily answered for this question today. So revision is very important. The scleroblast secrete the spicules. The spongin fibers, the spongioblast secreting the spongin fibers. These cells are important. Clear? Okay. Okay. Let's come back to the skeletal system of porifera. Almost all sponges is provided with a skeleton. Good. Found embedded in the mesenchyme. Okay. See, there is an outer pinacoderm and the inner coinoderm layer, but the skeletal system it is embedded in the mesenchymal or mesenchymal cells, which is present at the middle.
And what is the cells which is present in the mesenchyme or mesophyll cells? Just tell me. Few minutes before I explain this. What is the name of the cells? Outer layer pinacocytes, inner layer coinocyte cells, which has the flagella in it. And what is the cells, which is very good? Amoebocytes. Super. Now, this skeletal system is embedded in the mesenchyme cells. This may consist of separate spicules or of interlacing spongin fibers. See, with respect to the skeletal system of sponges under the porifera, the skeletal system is either made up of the spicules or it is made up of the spongin fibers. In some of the skeletal system of sponges, the skeletal system contains both the spicules as well as the spongin fibers. Clear? So the question may be asked, in every species of sponges in skeletal system, both the spicules as well as the spongin fibers has to be present. Then only that make up the skeletal system of the sponges. The statement is completely wrong. The skeletal system is either made up of spicules or made up of the spongin fibers, or it can be made up of both spicules and the spongin fibers. Clear? Next, this what is the purpose of skeletal system? It has to protect the body parts present in the organisms. Okay, the main function of the skeletal system is to protect the soft body. It's to protect the body parts of the organisms, and it gives rise to a framework for the organisms. Okay. Now, come back to the spicules and how these spicules look like and what are all the types of the spicules. And based on the types, we have named the structure. See, spicules are the crystalline structure consisting of the spines or the rays that radiate from the point. Okay, the spicule the spicules are the crystalline structure and it consists of the spines or rays. I'm going to say what is spines and what is rays. See, this spicules is secreted by the special mesenchymal amoebocytic cells. And what is the purpose of this amoebocytic cells is they gives rise to the scleroblast. And from this scleroblast, they are giving rise to the spicules. Don't forget the statement, very, very important statement. They are secreted by the mesenchymal amoebocytic cells, and this amoebocytes is going to produce the scleroblastic cells, and this scleroblast is the one who gives rise to the spicules. Okay, and some of the amoebocytic cells will give rise to the spongioblast, and that spongioblast gives rise to the spongin fibers. Okay, fine. And both scleroblast and spongioblast are coming out from the mesenchyme or produced by the amoebocytic cells, mesenchymal amoebocytes. Now, and what is this spicules made up of? We need to have the core organic material around which it is deposited either calcium carbonate or the colloidal silica. Either it is made up of the calcium or the silicon. Okay, calcite or the silicon. And these spicules are of two types, calcareous and the silicons. If the spicules is made up of calcium, then it is called as the calcareous. And those organisms and those species of sponges are placed under the class Calcarea. Most important statement. See, if the spicules of the sponges, it is made up of the calcium, then it is called as the calcareous sponges and those organisms which is placed under the class Calcarea. If the spicules, it is made upon by the silicon, if it is made by the silicon, then it is said to be as the silicaceous sponges, and that is based under the class Hexactinellida. So the question may be asked is the sponges which is placed under the class Hexactinellida, in that sponges the spicules is made up of silicon or the calcium. The question may be asked. So those sponges which is placed under the Hexactinellida, in that organisms or the sponges, the skeletal system is made up of the silicon. So it is a silicaceous sponges. Right. Next, according to the size according to their appearance, the name will be different. And see, according to the size, the larger spicules constituting the main skeleton, which is called as the megascleres, and the smaller spicules which occur interstitially, it is called as a microscleres. We know that spicules is secreted by the scleroblast. What is it? Scleroblast. If it has if it is a larger spicules, then it is called as the megascleres. If it is a smaller spicules, then it is called as the microscleres. Okay, these are all the structures. This spicules may be occur in different forms. One, it is simple rod-like structure may take the form of forks, anchors, shovels, stars, plums. They may be occur in different types, but that is the skeletal system of the sponges. Okay, these forms depends upon the number of axes and the rays. According to the number of axes and rays, these spicules has been divided into the different types of the skeletal system. And the first one is monaxon. Monaxon spicules are formed by the growth of one axis. If it has only one axis, then it is said to be as the monaxon. See, this is the point which have been erased. It has only one axis, one central axis. Then these are all the things which is called as the monaxon. These may be straight, needle-like or rod-like or maybe curved. Their ends may be pointed, knobbed or hooked. See, their ends may be pointed or knobbed or hooked. But there may be only one axis. Then only it is called as the monaxon. In monaxon, there are two different types. And what is that is monactinal and diactinal, in which growth takes place only in one direction only. That is said to be as monactinal, and the growth occurs in both the directions. Then it is said to be as the diactinal. See, first two is monactinal and the second one is diactinal, because the growth can be takes place from here also, and the growth has been takes place from here also. It has been occurs in the both the directions. So it is said to be as the diactinal. And in monaxon, the growth takes place only from here. Monactinal. In monactinal, the growth takes place from here only. And in here, the monactinal, sorry, the growth takes place only in one direction. That is why this is said to be as monactinal and this is said to be as the diactinal in nature. See, and one more important point is this monaxon can be either calcium, it is made up of a calcium or the silicon. So these monaxons are both calcareous and the silicaceous type. Okay, don't get confused. Next comes the tetraxon. If the spicules have the four rays, then it is said to be as the tetraxon, each pointing in different directions. Each pointing in the different direction. That is said to be as the tetraxon. Usually, one of the four rays is elongated, giving the appearance of a crown of three rays. It has appearance of crown of three rays. Such spicules is said to be as the trianes. See, usually one of the four rays is elongated. This is an ray, and one of the rays elongated, giving rise to appearance of the crown. For these three rays, this ray acts as a crown. That is why this structure is called as the trianes. Clear? However, when all the rays are equal, the spicule is termed as the calthropes. If all the rays like this, if all the rays are equal, then it is said to be as the calthropes. Okay. Now, when all the four rays persist, the tetraxon is also referred to as the tetraradiate or the quadriradiate. Just to see, just hear me what are all the things I'm explaining. At the end, I'm going to tell what are all the things you have to focus on. These are all unnecessary points, but you have to aware of these words. What is a rays and what is the axis like this? Fine. However, when one of the four rays is lost, the spicules become triradiate, which is the characteristics of the calcareous sponges. See, if it has the four rays, then it is said to be as the tetraxon. But here, when one of the four rays is lost, then the spicules becomes the triradiate. Then also it is considered as a tetraxon, because initially it has the four rays, but in the middle it has lost one of the rays. Then it is said to be as a triradiate, and this is the characteristic features of the calcareous sponges. If the elongated rays bears a disc at the both ends, it is said to be as the ampidistic, like this. It has the disc-like structures at both the ends, so it is called as the ampidisc. Clear? Next, triaxon. If the spicule has the three axes, then it is said to be as the triaxon. That crosses one another at right angles to produce 6 rays. They are considered, see, this is the right angle to this. 1 minute. And this axon is considered as the right angle to these. This is said to be as the right angle. So like this, one curve that crosses one another at the right angles to produce the 6 rays. This is what said to be as the triaxon. The question may be asked, in which type of axon the spicules cross one another at right angles to produce 6 rays? And you have to know this will be the triaxon. So it producing the 6 rays. As it is producing the 6 rays, it is called as the hexagonal. And this triaxon are the characteristics of glass sponges which is placed under the class hexactinellida only. So you have to know this triaxon is the characteristic features of only the glass sponges. That too, not the see. Remember the statement which I have been using. Triaxon are not the only characteristic features of the sponges which is placed under the class 2 hexactinellida. This statement is wrong, but the triaxons are the characteristics of glass sponges. See, there is also the other sponges which is placed under the class 2 hexactinellida, but only the glass sponges which is placed under the class hexactinellida, it is to be as a triaxon. Definitely, it will be the triaxon. So, see the difference between these two states. Okay. Next, polyaxon. These are the spicules with 7 equal rays. Spicules with 7 equal rays and radiating from the central point. Radiating from the central point, they may be grouped to give starfish appearance. And as it is have a different rays, it is called as the polyaxon. And this is the common among all the microscleres. Okay, fine. How the spicules have been formed. Above all these, these are all the things, the types of spicules, but now we are going to see about the development of the spicules. How the spicules have been arising. And name the cells which has, the spicules have been produced. What is the cell which gives rise to the spicules? Name the cells. What is the name of the cell? Quick. Lalita, Shanti Karanya. Sollu. Answer for this question. Name the cells. It is scleroblast. Scleroblast is the cells gives rise to the spicules. Spongioblast is the name of the cell gives rise to the spongin fibers. Okay, fine. How the spicules have been developed means, see this calcare. Let me explain you the picture first. See, initially this is considered as the mesenchymal scleroblast. See, this is the cells inside the structure which is called as the mesenchymal scleroblast. So, just remember the name of the cell which is gives rise to the spicule is scleroblast. Clear? And the scleroblast initially or the scleroblast generally it has two nucleus. It is binucleated in nature. Binucleus, it has a two nucleus. Between these two nucleus, calcium carbonate particle is present. See, what is the structure of this scleroblast? See, in the scleroblast which is present in the mesenchymal cells, It is two nucleus. It is binucleus in nature. It has a two nucleus within it. Between these two nucleus, the calcium carbonate particle of calcium carbonate is present. Initially, it is small, but later this calcium carbonate particle will be elongated inside the mesenchymal scleroblast. Now, this scleroblast will again elongate its structure and gives rise to a structure which is called as the spicule. So the elongation of the calcium carbonate particle gives rise to the spicule. Now see the nucleus, which is as initially it was in the binucleate form, one at the base and both at the middle, but later when the spicule is formed, one nucleus is at the base, one nucleus is at the top. Okay, now after some time, this spicule will move to the tip. This is the complete structure of the spicule. So you have to remember, what are all the things you have to remember is, this scleroblast is the binucleate structure. This scleroblast is binucleate. And initially, this scleroblast between these two nucleus, the spicules will be present here. Okay, and after some time, this it is a calcium carbonate particle, but after some time, this calcium carbonate will elongate the structure and gives rise to the structure called the spicule. And this spicule will reaches the tip. This is the correct structure of the spicule. When the spicule, after it reaches a certain stage, it moves at the tip and the nucleus will again come to the center point. Okay, fine. The calcareous spicule is secreted by the special scale which is called as the sclerocytes or the scleroblast. Okay, sclerocytes, which is derived from the mesenchymal scleroblast. The monoaxon spicule or the each race triradiate spicule is secreted by the group of sclerocytes, one acting as the thickener cells and the other acting as the founder cells. See, this is the spicule and the secreted, and this spicule secreted by the group of sclerocytes. The spicules which is secreted by the sclerocytes, one acting as the thickener cell and one acting as the founder cell. Fine. And now this scleroblast, as I said before, it is binucleate in nature, is believed. This is the cells. This binucleate, this nucleus cells is said to be, is believed to give rise to these type of thickener cells and the founder cells. Clear? And it is believed, it is not confirmed. Formation of spicule begins as a deposition of a particle of calcium carbonate between the two nuclei. Between these two nuclei, there is a structure, there is a particle which is made up of the calcium carbonate. Now this particle grows, drawing apart first the two nuclei and then the two sclerocytes. Thickener cells lays down additional, as the name suggests, this cell is called as the thickener cells. It going to add an extra calcium carbonate to the spicule. That is why it is called as the thickener cells. When the spicule is fully formed, both the cells wander into the mesenchyme, and the scleroblast secreting a calcareous spicule is called the calcoblast, and while the scleroblast secreting the silicaceous spicule is said to be a silicoblast. Clear? Now, the silicaceous monoaxon is believed to be secreted by the single silicoblast, while the six-raced triaxon is secreted by the nuclear division of the single silicoblast. See, if it want to give rise to the monoaxon spicules, then a single silicoblast is sufficient to give rise to the monoaxon. In case it needs to give rise to the triaxon, which has the 6 rays in it, it needs to divide first. This silicoblast undergoes the nuclear division, and after the nuclear division, it gives rise to the triaxon. Okay. Fine, that's all about the spicules, how the spicules have been developed. Now coming to the spongin fibers, how it looks like. This spongin is an organic. This is a proteinaceous compound. Okay, as this spicule is a calcareous or the silicaceous component, this spongin fibers is the proteinaceous substance. What I have to explain about the spicules? See, you have to remember when it comes to the skeletal system of sponges, you have to remember two things. See, Shanti Karanya, just take down what are all the things I'm saying now. Okay. See, when it comes to the skeletal system, when you read about the skeletal system of Porifera, you have to remember two things. The organisms which comes under the Porifera are the sponges. It is made up of two things. When it comes to the skeletal system of Porifera, it is made up of two things. One is the spicules or the spongin fibers. Okay, whether it is made up of either spicules or the spongin fibers, or it is made up of both spicules and spongin fibers. And the second thing, after the thing, what is the important thing is, see, this spicules is made up of the scleroblast. Just now I have explained how the scleroblasts have been explained. The spicules are made from the sclerocytes, or these sclerocytes will be derived from the mesenchymal scleroblast. Now, the spongin fibers, it is made from the what? Spongioblast or the spongiocytes. Spongiocytes. Okay. And after, see, first thing is very good. Spongioblast. Now, when it comes to the spicules, there are different types of spicules is present. This spicules is made up of either calcium or the silicon. The third thing, see, the first thing you have to remember is what is the skeletal system is comprised of, and this is the thing. Spicules and the spongin fibers. And the second point it is important is the spicules is made from the sclerocytes, and the sclerocytes is derived from the scleroblast. And the spongin fibers, it is made from the spongiocytes, and the spongiocytes is derived from the spongioblast. And this spicules is made up of, and the third important thing you have to remember is the spicules is made up of either calcium or the silicon. If it is made by the calcium, it is said to be as the calcareous spicules. If it is made from the silicon, then it is said to be as the silicaceous spicules. And now the spongin fibers. Spongin fibers, it is made up of the protein. Okay, proteinaceous spongin fibers. Okay, now coming from the spicules. The spicules is made up of two types. One is calcareous and silicon. I have explained this before. Now, there are different types of spicule is there. What is it? Monaxon, triaxon, tetraxon, like this. Polyaxon. If it has only one axis, then it is said to be as a monaxon. If it has three axon, that is said to be as a triaxon. If it has the four axon, then it is said to be as a tetraxon. If it has many axon, then it is said to be as the polyaxon. Okay, fine. If it has the three axon, then it has the six rays in it. Clear? If it has three axes from if it has the three axes from the center, it has been arrived, and here the growth has also occurs from here also. The growth has also occurs from here also. So the three, if it has the three axes, each axis has two growth points. Then it is produces the six rays. Okay, this is the difference between the axis and the rays. Like in the same way, everything will be applied. Okay, fine. This is about the axon. Now, coming to the development of the spicules. Look at the picture. This is the scleroblast. This is the mesenchymal scleroblast. And this mesenchymal scleroblast has the two nucleus in it. So, it is called as the binucleated mesenchymal scleroblast. Okay, so between these two nucleus, the small particle, it is made up of the a small particle is present here, and that particle is made up of the calcium carbonate. After some time, this calcium carbonate will be elongated. See, these two nucleus are going to secrete another two types of cells within the scleroblast, and the name of these two cells are thickener cells and the founder cells. Okay, and the thickener cells will add the extra calcium carbonate, will add the calcium carbonate to this small particle, and this tend to elongate. And after this elongation of calcium carbonate, this becomes the spicule. So this is why we are saying this spicule is made up of the calcium carbonate. Clear? And now see the structure. Now in between these two nucleus, the spicules have been formed. This one nucleus will give rise to the thickener cell, and another nucleus will give rise to the founder cell. See, now between these spicule, this founder cell is at the base and the thickener cell will be at the top. Now after the maturation of the spicule, again the nucleus will be near to each other. This is how the spicule will be formed. Okay, and now if the monaxon will be formed, a single spicules or the single silicoblast or the single calcium, it is the spicules which is made up of the calcium calcareous spicule. Single calcareous spicule is enough. And if it is the if the spicule is made up of calcium, a single silicoblast is enough to give rise to what? To give rise to monaxon. But if it wants to give rise to triaxon, which has the 6 rays in it, the scleroblast has to completely undergo the division, nuclear division. If it undergoes the nuclear division, there is many scleroblast is present. Now the scleroblast will give rise to the triaxon, tetraxon, polyaxon. That's all about the development of the spicules. Any doubts? Clear? Shanti Karanya, is it clear for you? If you have any doubts, you can ask me now. Okay. Lalita, clear for you? Okay. That's all about the spicules. Now we are going to see about the spongin fibers. This spongin fibers is made up of the protein. Okay, it is proteinaceous in nature. So this spongin is an organic, horny, elastic substances resembling silk in the chemical composition. Like nails, hairs, feathers, it is a scleroprotein containing the sulfur which is chemically related to the collagen, a horny protein. These are all said to be as the horny protein. Okay, see, this spongin is insoluble in nature. 1 minute. This spongin fiber is insoluble in nature. It is chemically inert and it is resistant to protein digesting enzymes. These are all the important things. It has the resistance against the protein digesting enzymes. Now, this spongin contains a large amount of iodine, reaching 8 to 13% in certain tropical species of the Spongidae and the Aphyllizinidae. These are all not important. What does spongin means? It is a protein. And what are all the things it possesses in it means iodine, and it is resistant to digestive enzymes, it is insoluble, it is chemically inert, and it is a horny protein. Okay, fine. Now, it is interesting to know that old herbs doctors for centuries used bathe sponge as a cure for croup, a throat condition of children resulting from the inflammation and particle obstruction of larynx. The most important point is this. So the sponges, from this point, you have to understand the spongin also has some of the economic importance, mainly medicinal importance. See, there is a type of sponge which is called as the bathed sponge that has a cure for the croup. And what is the croup means? This is the throat infection. throat condition of the children resulting from the inflammation and partial obstruction of larynx. Okay, this is an important point. Now, spongin occurs in various forms in the class Desmospongiae. In the class Desmospongiae, the spongin fibers will be in different forms, and it may be as a cement connecting together a silicaceous spicules as many monoxida. As I said before, in the class Desmospongiae, this poriferans or the sponges which possess both spicules and the spongin fibers in their skeletal system. Okay, in that skeletal system, the spongin is the proteinaceous compound which act like a cement, which connecting together the silicaceous spicules. Okay, fine. In human beings, consider the ligaments and tendons, which connects the bone to bone and bone to the muscle, like in the same way, which connects one spicules to another spicules by the spongin fibers. They act like a cement which connect one spicule to the another spicule. Clear? Fine. Next, more usually it is found in the form of branching or anatomosing fibers in which silicaceous spicules are embedded. This is called as the monaxinoideae. In keratozoa, these spicules are completely absent, spongin alone form. So what are all the things you have to remember from this point is same things I have been repeating again and again. So don't forget this point. The spongins, the skeletal system of sponges, either it is made up of a spicules, only spicules, either it is made up of the spongin fibers, that is only spicules or only spongin fibers. In some of the poriferans which is placed under the desmospongiae, in their skeletal system, it has both the spicules and the spongin fibers. Okay, but it is not necessary in the sponges. It has to present in the skeletal system, both spicules and sponges is present. Okay, you have to understand whether the spicule is present alone and the alone only spongin fibers is present. But in some of the poriferans which has both in their skeletal system, in that when the if the poriferans or the sponges which has the both the spicules and spongin fibers in their skeletal system, spongin fibers act as the cement which connects one spicule to the another spicule. This is the most important point. No other thing is important. And another point is important, which cell it gives rise to which type of skeletal system. Which, for example, spongioblast gives rise to the spongiocytes, and that is gives rise to the spongin fibers. If it is this scleroblast, it is gives rise to the sclerocytes, and this sclerocytes is the one gives rise to the spicules. Either it is made up of silicon or the calcium carbonate. Okay, fine. How the spongin fibers looks like and where it is have been developed from. See, it is secreted by a flask-shaped mesenchymal cells. Spongioblast. You have to remember this spongioblast is flask-shaped. See, both another point in you have to remember is it has a mesenchymal cells. In sponges we have a two different layer, outer layer and inner layer, outer pinnacoderm and the inner coinoderm, pinacocytic cells and the coinocytic cells. Between these two, mesenchymal cells is present. In mesenchymal cells, it has amoebocytic cells. These amoebocytic cells is the one gives rise to the two different types of cells. One is scleroblast and the spongioblast. Scleroblast and the spongioblast. This scleroblast gives rise to the spicules. Okay, and this spongioblast gives rise to the spongin fibers. And this spongin fibers is the one it looks like a flask shaped. Okay, you have to read like this. You have to make a flow chart like this. If you read like this means it will be very easy for you. See, this spongioblast is the flask shaped in it, and which are seen coating the fibers. That is why it is called as the spongin fibers. Now, the spongioblast becomes arranged in rows, and the spongin rod secreted by each fuses with those neighboring cells to form a long fiber. One spongin fibers will be connected to another spongin fiber, so it looks like it forms the long fiber. Now this spongioblast later became vacuolated. There is nothing present in it. That is why it is called as vacuolated. And degenerate after having secreted a certain quantity of spongin. If the scleroblast gives rise to the spongin fibers, and this scleroblast will become vacuolated because all the contents are involved in making the spongin fibers. So at the end, there is nothing resulted, nothing is there inside the spongioblast. So after some time, it becomes vacuolated and destroyed, and after it gives rise to certain quantity of the spongioblast. So what you have to remember is, if all the contents been gives have been involved in using the or producing the spongin fiber, there is nothing left in the spongioblast. So ultimately, it will be destroyed or degenerated. That's all about the skeletal system. Now, if you have any doubt, means you can ask me now. The only leftover part is reproduction. After finishing this reproduction, we are going to solve the questions. Okay, fine. Reproduction in sponges. We know both sexual reproduction is also seen in sponges, asexual reproduction is also seen in these sponges. First thing we are going to see is asexual reproduction. We know what is meant by asexual reproduction. There is no need of two individuals to give rise to the offspring or the progeny. One single individual, either it is whatever be the gender it is, generally it is, we can consider them as a female, because that gives rise to a next generation. So there is no involvement of two different gametes in the asexual reproduction. And what are all the types we have seen in the asexual reproduction means budding, fission, or the reduction bodies, gemmules. Okay, and first one is budding. In this method, the number of individuals is increased in sponge colonies or the new colonies or the developed. How it has been developed? Numerous archaeocytes collect at the surface and the pinacoderm bulges out to receive them. The bud so formed grows in an adult individual. It either remains attached with the parent individual or gets detached and attached nearby to develop into the independent colony. Let me explain you. See, this is the sponges. Okay, and to initiate the asexual reproduction, that too specifically the budding, the important cells required for this budding is archaeocytes. See, the name of the cells is important, like pinacocytes, coinocytes, sclerocytes, spongiocytes. Archaeocytes is also important. For what? Asexual reproduction, that too particularly budding. These archaeocytes collect at the surface at one end of the body of the sponges. In the pinacoderm, what is pinacoderm? Outer layer of the sponges is called as the pinacoderm. Now, this archaeocytes are being collected at the surface. The group of archaeocytes seen at the surface. Now, the pinacoderm, that is outer layer, when all the cells, that is when all the archaeocytes grouped at one end, how it looks like? The layer will be bulges out. See, and that bulge will give rise to a separate individual. At the end, this individual can be allowed to grow in their parent individual also by attaching with the parent also. But see, either it is allowed to grow continuously with its parent, also it can be separated from the parent and it can be attached with some other colony. Okay, this is how budding will occur. And what is the main important thing for to occur to budding is archaeocytes. The group, when the group of archaeocytes collected at one end, the pinacocyte starts to bulge and it gives rise to a separate sponge. And this sponge is now allowed to grow with its parent also. Also, it can be separated from the parent. It can grow as a colony or even an individual. Okay, fine. This is about budding. Next comes the fission. In some sponges, multiplication takes place by the fission. That too, which type of fission? It may be the binary fission or the multiple fission, throwing off the parts of the body. This sponge is hypertrophied over a limited area, developing a line of weakness. Along this weakness line, splitting occurs and a part is separated from the main parental body, which develops into a new individual. By budding, this new individual takes the form of a colony. See, in the fission, we know splitting will occur. Sponge is split or broken into a pieces. See, this is a full parent sponge, a completely developed or a mature sponge. In that sponge, the splitting is occurred. The area where the splitting is occurred, this is said to be as a line of weakness. There, the cells have been become weak, and after it has forms a splitting. Each split gives rise to a separate individual. These are all the parts of the parental body, the parts which have been thrown out from the parental body, okay, by accidentally or purposely. Okay, if it is a purpose, then it is said to be as a fission. If it is an accident, then it will be said to be as a fragmentation. Anyhow, they are going to develop into a new individual. That is why I have said you these sponges have an extraordinary regenerative capacity. When you read about the general characteristics, it now you have to connect the point. Now you have to connect the point where we have read in the general characteristics. There we have read, even if we cut the sponges into different minute species, minute parts, they can give rise, they have the capacity to regenerate and gives rise to a single individual. That is why reading the general characteristics is very much important. That this is how you have to read, by linking the characteristics. Okay, if it is by accident, then we can said to be as the fragmentation. And if it is purposely it want to gives rise to many, it has to multiply its colony, then it is said to be as the fission. Okay, fine. That's all about the fission reaction. Now, what is the formation of reduction bodies? Another very unusual method. This is not an usual method. This is an unusual method of asexual reproduction, is the formation of the reduction bodies. Many freshwater and marine sponges disintegrate in adverse circumstances. Favorite conditions. Unfavorable conditions. Ailing sponge will usually collapse, leaving some small rounded balls, which is called as the reduction bodies, and each body consists of an internal mass of amoebocytes. See, what are all the important cells seen in this type of reproduction system? A amoebocytes covered externally by the pinnacle. When favorable conditions return, these reduction bodies goes to a complete sponges. See, this is the correct sponge, mature sponge. See how it looks like. There is a spicule is there. There is an amoebocytes. Amoebocytes is there. This is an outer layer pinnacle. Now, during an adverse condition, during an adverse condition, these sponge will collapse, and they are looks like a small rounded bodies, and these rounded bodies is called as the reduction bodies. In adverse condition, these sponges will alter its structure and gives rise to a small rounded balls like structure, and that small rounded like structure is called as the reduction bodies. They have amoebocytes within it, and covered, these amoebocytes will be covered externally by the pinnacle. When the favorable conditions will occur, this each reduction bodies gives rise to a new sponge. Okay, this is about the reduction bodies. Fine. Next, formation of the gemmules. All the freshwater and some marine sponges have a regular and peculiar mode of asexual reproduction by the internal buds, that is called as the gemmules. The word gemma is derived from the Greek word which means the bud. Okay, see the same things like budding. Okay, these eventually detach and develops into a new individual. See, have a regular and peculiar mode of regular reproduction by internal buds. These are all the internal buds. They have been covered by a covering, and between these, this is the mesenchymal cells. There, the spicules will be present, and after that, it is covered by the pinnacle. Okay, and each buds gives rise to each individual. Gemmules enable the sponge to tide on unfavorable condition, such as excessive cold or drought, because they can withstand freely and considerably greater degrees of desiccation than do the adult sponges. Consider the cyst. During unfavorable condition, the organisms or the protozoans undergo a cyst. It has been enclosed by an extra wall called the cyst wall, which is the process is called as encystment. The same thing followed by the spores, that is called as the formation of gemmules. During unfavorable condition or adverse condition, they tie they make this sponges to covered by an extra covering, that is called as the gemmules, and if it forms the gemmule like structure uh, during an adverse condition called the cold or the drought, it can easily withstand. But during the favorable conditions are return, these gemmules, each internal bud which is covered by the pinacoda will be released into one new sponges. A full-grown gemmule is a small heart consisting of an internal mass of food-bearing arcuocytes. So you have to understand what is the function of the arcuocyte is. It has a lot of food within it. See, during a unfavorable condition, it forms a gemmule-like structure. In that structure, it has to withstand freely. Further, it needs food, and that food will be stored within the cells called the arcuocytes. Okay, and this archaeocytes will be covered by the resistant chitinous covering so that it can prevent themselves from an external danger. A chitinous covering or an envelope or capsule, and usually opened at an end by an outlet called the micropyle. See, this is an opening which is called as the micropyle. And the protecting envelope may be strengthened with silicon amphidisc spicules euphydacea or the monaxon spicules spongilia. See, these are all the examples. You have to remember the examples. If it is an amphidisc spicules, the spicule which is covered like this at the both ends, this is said to be as the amphidistic. And monaxon spicules, you know that this is the monaxon spicules, and example for this monaxon spicule is spongilia. So the spongilia and the euphydacea, they are going to form the gemmules. Now, in autumn, the freshwater sponges suffering in cold and food scarcity die and disintegrate, leaving behind the larger number of gemmules. Now, after receiving the a large number of gemmules, they are going, which remain inert throughout the winter because it is in the gemmule form. Now, as the base of the parent sponge decays, the gemmules are set free, and these either sink to the bottom or simply float away. In April or May, when the conditions again become suitable, these gemmules begin to hatch. That is why this gemmules is said to be as the perfect asexual reproduction method. Their living contains histioblasts. This is nothing but these are the things gives rise to the tissues. Escape through the micropyle. Micropyle is the opening and develop into the new sponges. During adverse condition, it forms the gemmule-like structure. Okay, which contains a lot of internal buds. And after that, during favorable conditions will occur, the parent will disintegrate and all the internal buds will give rise to an each sponge. Okay, these produces ova or spermatozoa, which gives rise to a summer generation of these sponges, which in turn die away in atom, leaving behind the gemmules. The life history of such sponges thus illustrates an alternation of generations. Okay, alternation. So this is the most important point, which they have given the list of phylums, phylum Porifera, Coelenterata, Annelida, Arthropoda, like this. Which are of the phylum gives rise to an alternation of generations? They can give a statement like this, and this alternation of generation will be explained very clearly when I'm going to teach the Coelenterata. Okay, just now, for now, you have to remember the sponges can also exhibit an alternation of generation. Okay, and if you have any doubt means you can ask me now. That's all about the asexual reproduction. Any doubts? Tell me fast, quick. Okay. Okay. So just remember, let me tell me the simple word and we'll finish this asexual reproduction. Just remember the alternation of generation means they can give, they can undergo a different forms of life stages, which means for some time it undergoing a sporocyte form. See, these gemmules are nothing but the spores. In unfavorable condition, they are undergoing in a cyst form. Their life stages in this spore form will be remain the same. The food will present inside the archaeocytes and keep on take the food from the archaeocytes. During the unfavorable condition, it come back to the normal lifestyle. So this alternation of generation from the normal lifestyle and from the spore life stages. This alternation of generation from the normal to the spore form, it is called as the alternation of generation. So you have to remember Porifera also has the capacity to exhibit this alternation of generation form. Now comes to the sexual reproduction form. So you have to know what is the meaning of hermaphrodite and what is mean by monoecious and what is dioecious. Anyone remember what is monoecious, what is dioecious, what is hermaphrodite? If you know means just text me. Just 2 pages is remaining. We'll finish and go to the questions. See, most sponges are monoecious, that is hermaphrodite, but dioecious unisexual sponge species also known. Which means what? See, first let me explain what is monoecious. Monoecious means both male and female reproductive organ will be present in the same individual. What is it? Both male and female gamete, both female and the male reproducing organ will be present in the same individual. That is said to be as monoecious. This monoecious is also said to be, is also called as a hermaphrodite animal. Just make a note of it. The question from this word can also be asked in your exam. Let me prove to you when I'm showing the questions. See, most sponges are monoecious. Monoecious means what? The sexes are not separate, which means what? The both male and female sex organs will be present in the same individual. In case it is a dioecious, female it will be the separate individual, male will be the separate individual. They individually gives rise to their own gamete and finally they are going to fuse together to fertilization will occur. After the fertilization, at the end of fertilization, zygote will occur and the sexual reproduction will continue. But in case of monoecious, both male and female will present in the same individual. In case of dioecious, male will be different, female will be different. Okay, that is unisexual. Only one sex is present in the each individual. In case of monoecious, it is bisexual. See the term monoecious, bisexual, hermaphrodite. All the terms are similar. In case of dioecious, only the sexes are separate. Sexes are separate. Here, sexes are not separate. Both are present in the same individual. But here, they are separate individual. It is said to be as the dioecious. Clear? Any doubts? No. The sperms and the ova are derived from the archaeocytes and the choanocytes which undergo the gametogenesis. See, the sperms and the ova are derived from the archaeocytes or the choanocytes which undergo the gametogenesis. Which means what? What are all the cells again come into a picture? Archaeocytes are the choanocytes. They are the one gives rise to, they derived from the, they are going to give rise to the sperm or ova. Okay, then again, so they are undergoing the gamete synthesis, that is gametogenesis. Although most sponges are hermaphrodite, but cross fertilization is the rule. What is it? In hermaphrodite, both the sexes are present in here. There is no need of cross fertilization seen in here. But what happening here? Although most sponges are hermaphrodite, which means what? Both male and female. See, what is cross fertilization? This is a male. Consider this as a male. This is a female. This is male and this is the female. This will be produced male gamete. This will produce the female gamete, and they are going to fuse. This is called as the cross fertilization. But most of the sponges are the hermaphrodite, which means what? Both male and female will present in the same individual. Of course, it will be the same, the male and female sex organs will be present in the same individual. They are preferred to cross fertilize with the other individual. Why is it so? Because eggs and sperms reproduce at the different times. The time period, the eggs, see, of course, they are cells which gives rise to the male sex organ and the female, that is eggs and the sperms. The timing will be different. Okay, if the female gamete is ready to fertilize, the male is not synthesized yet. So obviously, this female will prefer other male sex organs to fuse. This is called as a cross fertilization. So you have to know, although most of the sponges is hermaphrodite, they prefer to cross fertilize because the time period of producing these eggs and the sperms will be different. Okay. Now, what is fertilization? The fertilization seen in sponges is external fertilization. What is meant by external fertilization and internal fertilization? I have explained right from the when I explained the general characteristics. Yesterday, I have explained what is external and internal fertilization. Let me explain once again. In case of external fertilization, the female will drop its egg in the water and the male will reach the female gamete by the pheromones. Okay, that will be the different part. Anyhow, it is going to reach the female gamete. Now it is ready to fuse outside the body of the individual. That is said to be as the external fertilization. Very good. Exactly. Outside of the body. And if in case of internal fertilization, it will be the development of the embryo will occur within the body of the female individual. Okay, now here the same thing as it is the female, as it is an external fertilization, the sperms are released outside through the outflowing water, make their way into the another sponge along with the increasing water current. Now the coinocyte, if it has to reach from one place to another, obviously which cell has flagella in it? Yesterday I will make you very clear which cell has flagella in it. Coinocyte has the flagella in it because by beating of this flagella, yes, very good, coinocytes. Because of the beating of the flagella, water current will be produced so that the male gamete will be reaching the another branch. So now this coinocyte acting as the nurse cells transport the sperms with the tails left being to the mature ova which lie behind the flagellated coinoderm. Fertilization takes place in situ. Clear? Now, development. How the development will be occur? There may be direct development and the indirect development. In case of indirect development, the young ones will undergo many of the larval stages and those larval stages will give rise to an adult form. In case it is a direct development, straight away the young ones will develop, they will grow in size and gives rise to an adult form. If the larval stages is involved in the metamorphosis stage or in the life cycle, then it is said to be as the indirect development. Okay, if there is no larval stages in seed, straight away the young ones gives rise to an adult just by increasing their size, means this will be the direct development. Clear? And what are all the larval stages? See, larval stages is very crucial. Just make a note of it. Take down your note and pen and write this. Amphiblastula and parenchyma are the larval stages of the sponges. Most important. There is no doubt they can ask the question. Okay, amphiblastula and the parenchyma. This is the structure of amphiblastula and parenchyma. It is a hollow oval larval stage. It is the characteristics of many calcareous sponges. Even if you don't know what is the class, just remember amphiblastula and parenchyma are the larval stages of the sponges. I'm repeating, don't neglect these type of questions. If you know, definitely here one question is there in the question in the exam, means you have to read that. Don't take any chance. No. Anterior one half of amphiblastula bears the flagella, and where the posterior half does not have any flagella. Just These are all the unique characteristics. In Amphiblastula, it is hollow or oval. Oval larval stage, it is characteristics of calcareous sponges. See, one half of the structure will possess the flagella and remaining half will not. Okay, these are all the unique characteristics you have to remember. In parenchyma, it is solid, oval, and flattened larval stage. It is a characteristics of some calcarea, hexactinella, and most desmospongy. Its entire outer structure bears the flagella. Okay, this is the flagella layer. The entire surface has the flagella. This is the difference between the parenchyma and the amphiblastula. If one half of the structure has flagella and others not, means then it is said to be as the amphiblastula. If all the body is covered by the flagella, means then it is parenchyma. And it is the characteristics of most desmospongiae, some calcareae, and some hexactinellidae. Okay, its entire outer surface bears a flagella. With the help of this flagella, the motile larvae escapes from the parental body and swim for a few hours to many days. Finally, they settle down, become attached to some solid object, metamorphosis, and grown into an adult. Okay, so this type of development is called as indirect development because it undergoes some of the larval stages and it grows to an adult. That's all about the reproduction. Any doubts with respect up to this? If you have any doubt, means we'll go to the questions. If you have any doubt, means just text me. Quick. Any doubts? Okay. Okay. Say see how the question have been arised here. Hermaphroditism in humans is a condition which is characterized by. What is hermaphroditism? Absence of gonads, loss of speech and vision, absence of external genitals, discrepancy between the gonads and the external genitals. Very good. Both sex having in the same individual. So, what will be the option? Very good. D will be the correct answer. See, we were studying this hermaphroditism in where? While studying about the porifera. But how the question will be asked in humans, whatever be the question, if you know what is hermaphroditism, at least you can eliminate the options. There is nothing called absence of gonads. Both will be present. There is no relation between the loss of vision and the speech with respect to hermaphroditism. There is no absence of external genitals. Both will be present. Only thing is there is discrepancy between the gonads and external genitals. So the answer will be option 4. Okay, fine. Next. What is the correct statement with respect to the canal system in sponges? In yesterday's class, we have completely seen about the canal system. See what are all the options here. Ascanotype is the most complex grade of canal system. Ascanotype is the most simple grade of canal system. And cycanoid is evolved from the ascanoid system, and the leuconoid is derived from the ascanoid system. Each and every point will be explained there. First of all, tell me which is the most simple type. Obviously, ascanoid type is the most simple type of canal system. Clear? And first point, ascanoid type is the most complex grade. Which is the most complex grade of canal system? Leuconoid type is the most complex grade of canal system. So obviously option will be 1 will be eliminated, 2 will be correct. So C and D will not come, only A and B. And the cycanoid type is evolved from the Cycanoid type will be evolved from the ascanoid system, and leuconoid type will be evolved from the ascanoid system. This point also I have explained multiple times. See, there is a different types: ascanoid type, cycanoid type, and leucoid type. This is the most simple type, this is the most complex, and this is the complex. This from this ascon type, the cycon type will be evolved. Okay, from this cycon type, this leucon type will be evolved. If we know this basic difference, you can easily answer this question. Okay, so the answer is the cycon type is evolved from the ascanoid system. In yesterday's slide, I have very clearly said those are all the poriferans which have the cycon type of canal system. Definitely in this larval stages, the larval stages of this particular poriferans, definitely they possess the ascon type. Okay, clear, Shanti Karunya? So the correct answer will be option 2. That is why revising is very, very important. These are all the basic questions. If you made mistakes in this type of question, means how will you answer those difficult questions? See, 2 questions have been resembled in this from the Porifera, and I will show you one of the questions. If you read the topic, means see how very easy to you to answer these type of questions. Another questions they have explained is, see, there is a comparison between these two. What is it? Phylum Porifera and the phylum Coelenterata. When we complete the phylum Coelenterata, then we will answer this type of questions. Okay, see how the question may be. That is why I keep on stressing why studying the general characters is very important. Okay, see, from the general characteristic they have asking the questions. Okay, so finally 3 questions are from Porifera. See how from very simple topic 3 questions. Fine. Okay, answer for this question. Which of the following is true about sponges? Innumerable mouth and one exit, one mouth and innumerable exit. Spicles are made of chitin. A large spacious stomach, which is true. Innumerable mouth. Mouth is nothing but ostia. Exit means osculum. See. Oh, you're confusing about the large spacious stomach and the first option. See, pores is said to be as the ostia. That is called the mouth. Okay, I have said there are more ostia, more pores is present, but only one osculum is present. when I'm explaining the canal system, I have explained this. Pores is nothing but the technical term of pores is ostia, and only one osculum is present. That is why the spongiocele, that is central body cavity, has become very slow when sending out the water through the osculum. So obviously the answer will be only one exit is there, only one osculum is there, one way of exit is there, but innumerable number of more, that is more pores is present in the body of the sponges. So the exact option answer for this question is first option, innumerable mouth and one exit is present. Clear? Answer for this question is option A. Fine. Next, flagellated chambers are found in. One minute. See, the answer will also be have been attached here. You can make use of it, okay? While you study separately, you can make use of the answer also. I'll post it in the group, so you have to. Okay, what is the answer? Flagellated chambers. This flagellated chambers I have explained when I studied about the. What is flagellated chambers? Just tell me what is flagellated chambers. Yesterday I have explained. Flagellated chambers is nothing but which canal is said to be as a flagellated chamber? Flagellated chambers. See, flagellated chambers are small, spherical, lined by the coinocytes. Yesterday's class I have explained multiple times. Radial canals have the flagella, so it is called as the flagellated canals. And incurrent canal are devoid of this flagella. Okay, this is the difference between these two. Radial canals, so it is also called as a flagellated canals. And where we have seen this leucon type. Okay, so again, revision comes into picture. So the answer for this question is leucosolenia. Of course, it is leucon type, but the question has here is found in leucosolenia. Fine. Parazoology is the study of. To answer this question, general classification is important. Okay. Initially, the animal kingdom will be classified into two. One is protozoa and the eumetazoa. In the eumetazoa, there are three things will be present, and one of the phylum is parazoa. The example for the parazoa, the phylum which is placed in the parazoa is Porifera. So, if you know the general classification of animal kingdom, you could have easily answered for this type of question. So, don't take lightly about the general characters and classification. If you didn't even know about the content, you can easily answer the question when you know the general characters and classification. So, the answer for this question is Porifera. Internal budding in some sponges is for. What is the purpose of budding? Reproduction, surviving during the adverse condition, asexual reproduction, growth. Asexual reproduction. What about Shantikaranya? Just now I have explained, right? See, internal budding. They are the type of asexual reproduction. Okay, common bait sponge is Spongia, Spongilla, Cleona, Euplectella. Right or wrong, just answer the question. Common bait sponge is the answer for a common bait sponge is Spongia. Okay, don't worry. Of course, it is not Spongilla. The answer for this question is Spongia. Okay, and body organization in sponges is it is a protoplasmic, tissue grade, cellular grade, or organ grade. Very good. It is a cellular grade of organization. Okay, fine. Costliest. The costliest Turkish baked sponge is Spongia officinalis, Hippospongiae, Spongilla, and Grantia. Of course, I didn't even teach these type of question, but these are all the factual information you should know. Okay. And the answer for this question, the costliest Turkish baked sponge is Spongia officinalis. Very good. Spongia officinalis. Paragastric cavity. What is paragastric cavity? It is a spongiocele, the central body cavity, and it is found in Leucosolinia only, all sponges, marine sponges, freshwater sponges. Paragastric cavity. If you know the general canal system structure, you could have easily answered for this question. Kindly answer with question number so that I can answer, I can comment to know for what question you are answering. Very good. These are the basic parts of the canal system. That is why I have explained what are all the parts of canal system. Of course, the canal system is the only characteristics of sponges. The paragastric cavity or this sponge or cell definitely is going to present in all the sponges. So no doubt it is found in all the sponges. Clear? Next. Sponges differ from other metazoans in having numerous mouthlets, one exit, both, none. Okay. So you two are confusing with the what I have is said there are numerous mouthlets. Of course, there are numerous mouthlets is there. That is numerous ostia, numerous mouth where the water can be easily enter into the body of the sponges. But there is only one exit is there, that is osculum. Then how the sponges differ from other metazoans is, of course, all the sponges, only sponges have numerous mouthlets and one exit. So both the structure has the unique characteristics. So this sponges will be differ from other metazoans in numerous mouthlets as well as the one exit is there. Yes. Okay. Clear now? Now. Next, sponges can be distinguished from other animals in having the spicules, water circulating system, choanocytes, and all of these. Okay, good, super. Next, Lalita. Very good. The answer for this question is all of these. Spicule is present. What is water circulating system? Nothing but water canal system. Canal system is said to be as the water circulating system. And choanocytes, which has the collar cells, are the very good canal. And choanocytes have flagella in it. Choanocytic cells is also called as a collar cell. So all these structures are the distinguished characteristics present only in the sponges. Now, sponges are characterized by choanocytes, canal system, innumerable mouthlets, and one exit, all of these. Very good. Superb. All the structures are characterized by the sponges. Now, the type of canal system exhibited by the desmospongiae. Again, for this type of question, you have to revise. Any guesses? Is it so? No, it is cycon. Okay, cycon type of canal system exhibited only by the desmospongia. Just remember, okay? No worries. Ascon type of canal system is found in. Ascon type of canal system is found in. So this is why I'm stressing type of canal system and perfect examples of canal system. So the Ascon type of canal system is found in. Any guesses? Let me say, Ascon type of canal system is found in the Leucosolini. Leucosolina. Okay. Now, which one of the following is the most distinctive characters of these sponges? Which one of the following is the most distinctive characters of sponges? They are acellular. They possess the special cells called coinocytes. They reproduce asexually. They are all marine. The question they have been asked is most distinctive characteristics of sponges. Acellular, coinocyte cells, asexual reproduction, they are all marine. A or B? See, let me explain. Acellular. Acellular means there are many organisms which have an acellular. So this is not the distinctive characteristics. Okay. And many organisms can reproduce asexual reproduction, not only sponges. And the sponges that they are all marine. Sponges are not all marine. They have the marine sponges is there, freshwater is there. So this three will not be the answer. These are all the not distinctive characteristics. But instead, it is a coinocytic cells. It is made up of the coinocytic cells. So see the general character, how the general characters are helping in answering these type of questions. Now, the sponge hexactinellidae lives in. I have explained when I have explained the general characteristics of each class. Calcarea, Desmospongia, hexactinellida. This is small, this is moderate, this is large. This is lives in the shallow, this is lives in the deep, and this hexactinellida is in both. Like that, I have explained you very clearly. And what does it means? It is in the brackish water. Okay, it lives in the brackish water. Okay, don't worry, I didn't explain this brackish water. Also, it has the capacity to live in deep or shallow. Just verify in the PPT. Okay. Now, indicate the freshwater sponge from the following assemblage. Okay. Let me go through only the simple, important questions. Just tell me the cells surrounding the osculum in sponges. I have explained you this multiple times. The cells surrounding the osculum in sponges is very good. You are saying perfectly incorrect answer. See, the cells surrounding the osculum in sponges is myocytes. Okay. See, the cells surrounding the osculum, one more part is there. The cells surrounding dash in sponges is again myocytes. Just refer that in the PPT. Okay. And now an example. Okay. Glass sponges belong to the class. Glass sponges belongs to the class. Glass sponges belong to the class. Very good. Hexactinellide. See, important question is this. See, what is the middle layer of the body wall of the porifera is in? Don't make a mistake in this question. Very good. Mesenchyme cells. Now, wait. Who describes the animal nature of Porifera? Robert Grant coined the term Porifera, but the Elias is the one described the animal nature of the Porifera. That is why reading the question twice is important. Okay, Robert Grant coined the term Porifera. Okay, Elias is the one who describes the animal nature of Porifera. Clear? Okay. Fertilization of sponge is. Fertilization of sponge is. What I have explained just before, it is external fertilization. Okay, the fertilization takes place outside of the body of the organism, external fertilization. Remaining questions you have to solve it on your own, and then tomorrow's class I'm going to start the sealant data. Okay, any doubts? If you have any doubts means you can ask me now or you can leave the class. Thank you. Tomorrow same time we'll meet you again. Tomorrow we're going to start the sealant data. Thank you so much. Thank you.